Looks like most of you have stopped writing, just a couple people, looks like they're finishing up, writing to the side. So, number one, how do you think the body repairs this injury? What do you think it does? Emily?
Well, you know, platelets will, like, create a barrier and, like, heal up your skin so that you actually stop bleeding. That's the scab that you get. But beyond that, I don't really know. I mean, I know platelets repair you, but further on, I don't know, like.
Okay. The platelets don't repair, but they do come in and stop. They clot the blood.
Yes.
And then yes, that's what when the blood lingers, you either have a bruise or it does help with the scabbing process. You're right. Then what? What do you guys think? What do you think after that? Obviously, we've got to have some sort of stop the blood if it's down in the bleeding or down in the tissue where it bleeds. But what if it even isn't that deep and it doesn't bleed and it's just a small one at the top and it doesn't go deeper into the nervous tissue and the dense connective tissue where the blood is?
Then the skin cells can start patching it up.
Yeah, the skin cells start to repair. Okay. How long do you think this process continues? How long do you think the skin repairs itself? Garrett, you're looking at me like maybe you want to give an answer.
3 weeks.
Maybe 3 weeks. Okay. Anybody else want to gather a guess? Emily?
I mean, most cuts or scrapes only take a few days really to be repaired.
Okay. So maybe we could tag on to the end.
The bigger the injury, the longer it would take. You know, if you get like a bullet wound, that's gonna take longer than a paper cut.
Right. So the amount of damage, it just depends on the amount of damage.
Well,
Sounds
it
good to me.
depends on your age too.
Yep. Very good.
Yes.
Okay, number 3 then. What do you think causes these cells to stop this repair process? Why don't they just keep on going? Make a big old blob that just keeps like forming on the outside of wherever that cut or that injury was.
They have restrictions in place and they understand that once this cut's healed, we're done and we don't have to do anything anymore.
Okay. Yeah. So basically there's got to be something in the organism that says, all right, it's time to stop. This is good enough. We're repaired. We're healed. Because if we keep going, we're going to look abnormal, right? So today, that's our whole point. We are going to try to figure out what are these things that cause the cells to stop replicating, to stop making new cells and say, hey, all right, we're good. We're repaired. Okay. So with that being said, we are gonna. This is just kind of an outline of what the notes are gonna look like for today. So you don't have to take this down because you're gonna be actually doing that anyway. All right. So what we're gonna take a look at first are what are some of the controls on cell division, and then what are some of those cell cycle regulators called. We have external regulators and we have internal regulators. And then what do we call it when there is uncontrolled cell growth, when we can't control the cell division? Okay, so we're going to start, like you said, again with some controls on cell division for today. All right, so first things first, when we are controlling the cell division, there has to be some external factors that basically give us some signals that say, all right, keep growing, or hey, we need to stop. So these are going to be physical. So what do we mean by physical? If it's a physical factor, it's something that does what to the cell?
It like, not like tells them, but like makes them stop.
Okay, but why? If it's physical, what's physical versus chemical? Because notice up there it says physical and chemical signals. So if it's a physical signal, then it's something that's doing what to the cell? Nathan, you look like you want to answer. If you get physical with someone, what are you doing?
Would it eat the cell? Would it kill the cell?
Well, it could, but what does physical mean? That's where we're trying to get. Like, if I am physical with Katie over here, if I get physical with her, what am I doing?
You're touching them?
Ding, touching her, right? I would go touch her. If I get physical and I show her, like I'm touching her. Okay, a chemical signal is a it's a what?
Non-touching?
Well, it could still touch, but it is instead of somebody touching, it might be an actual
Message?
Like signal through like chemicals in the cell.
yeah, it's a message, but it's utilizing chemicals to send that message to say, hey, all right, this is what's happening. And then the last type of external growth factor is, external factor is a growth factor. Okay, there are specific growth factors. Do you guys know what those growth factors typically are in our bodies? Starts with an H.
Hormones.
Hormones, yeah. So hormones might tell us, hey, yeah, keep growing or maybe stop. All right, so let's take a look at those. All right, so the physical one that we just said was when cells touch other cells. There's that physical component. So in this petri dish, and this is true story in my biochemistry class in college, we grew all kinds of great stuff in petri dishes. And yes, that's how cells are started. Like they will take human embryonic tissue and they will grow an ear in a petri dish. It's crazy, right? But it's true. These things happen in scientific laboratories. So when cells are in this petri dish and they touch each other, they may then send a signal that says stop growing. So I kind of did that in the petri dish. If the cells keep growing and growing and growing and growing, if they feel a cell on all of the sides of the cell, then that might be a signal to say, hey, stop growing. Okay? Again, the reason why this happens is not necessarily known, but there are some sort of receptors that are on the outside of these cells that when it touches another cell, those receptors get pressed and then those send the message internally to the cell that says, all right, we've got to stop growing. And so, it just keeps going and going. And then, other people ask, well, why don't these cells just keep growing and come out of the petri dish? Because again, I've witnessed this with my own eyes. These cells keep growing, but they don't grow outside the petri dish. They don't keep going up and over and spill out. They stop. So there must be some sort of receptor on the outside of that cell. When it touches something or touches another cell, it says, all right, it's time to stop. It's time to stop replicating. Okay. So again, I just kind of gave you that hypothesis, but you guys give me a good hypothesis here. If we're going to make a hypothesis about cells touching other cells, how can we put that in a good hypothesis form? I like to do this now just because, in the spring we're going to have that assessment that you guys all have to take, and you're going to have to know how to write a good hypothesis. Emily.
If a cell touches another cell, then it will know to stop growing.
Very good. Good hypothesis. All right. Now, internal factors. You heard me say, well, these external factors, these receptors pick up this information and then it's transmitted to the internal factors. And we are going to talk about those internal factors. What are they then? Because obviously the outside have to pick up some sort of signal and then they go into an internal factor that has to be relayed to the nucleus where all the information, the DNA, which is telling us what to do, says, hey, we need to stop. So we're going to get there. Okay, the first internal factor is what we call cyclin. It's great because we're in the cell cycle, right? So it's good to hear something that's sounds like it should go with the cell cycle. Cyclin. Okay, cyclin is a protein that regulates the timing of the cell cycle in a eukaryotic cell. So cyclin's kind of like the bells here in our school, okay? So without the bells, we would kind of be at a loss, oh, is it time to go? We could watch the clock, but there's nothing like signaling to us, all right, it's time to move to another class. It's time to leave this class. Okay, so cyclin's doing that in the cell, saying, hey, it's time to start replicating, or hey, it's time to stop replicating. Okay, and those external factors, i.e. the principles or the administrators here at school, they decide what that cell cycle is, or what our schedule here at school is, okay? The external factors obviously help to signal to the cyclin to say, all right, it's time to stop. We're touching something, time to stop, all right? Class is up, time is of the essence, all right, time to move to a different class. So kind of like that same thing. Okay, all right, so again, I put up this marshmallow looking thing because when you look at cyclin, it just looks like a small little particle inside the cell. You may not even be able to recognize what it looks like. All right, kinase, okay? Kinase, now, do you guys remember in first semester, what did we learn about the suffix ase? It indicates it's a what?
Enzyme.
Very good. And then remember, what do enzymes do for our metabolism, so to speak?
Speed it up.
Speed up our chemical reactions by lowering the energy required, right? So kinase is an enzyme, and it is activated by cyclins. So again, the cyclins are what's controlling this cell division internally, okay? How does it do it? It activates kinase, okay? And when kinase is activated, it transfers a phosphate group from one molecule to a specific target molecule. Remember when we talked about the active site? All right, and what does the active site combine with? Started with an S because it was called an enzyme blank complex. And the enzyme is what has the active site. Anybody remember? Anybody at home remember? I'm not up there, so you'll have to turn on your microphone. Nobody. Okay, substrate. Substrate, remember enzyme substrate complex, because the substrate has to be specific for that active site. So in this case, there's a phosphate group that gets transferred that acts as that substrate. And so it goes and it says, all right, we're gonna target this cell, this specific cell. Maybe it's a cell in your liver, okay? Because you've been taking medication and your body has a buildup of medication, and so your liver is gonna, those liver cells will help break down the harmful components of that chemical that you put into your body. And so in this case, kinase would be activated to go and break down that medication because the phosphate group was transferred to that particular liver cell. Okay, so go ahead and put that there. We've been talking. So what happens then, the energy is increased on that target molecule and it changes its shape, and voila, we start to have that cycle started, or in this case it could stop it. So cyclin activating kinase will decide if we're gonna start the cell cycling or if we're going to stop it cycling. Okay, everybody good with the external and internal controls? Great. Okay, so there is something, and this is kind of like the best place to put this, something called apoptosis. All right, and apoptosis is a programmed cell death. So yes, some of your cells have a programmed death that they're supposed to die in a specific time period. Okay, that it's only active for so many days and then boom, done. A good example of this would be your hair. Okay, your hair will grow and grow and grow, and then eventually that particular hair will fall out and it will die, and then a new hair will continue to grow. Now, for however many cycles this can happen is again based on genetics and sometimes outside factors like drugs and alcohol and things like that. So your body has
years, because of your cell deaths, your skin has like completely changed between those 4 years.
Right. Yes, and again, your skin is constantly being moved up and sloughed off. So yeah, you could, depending on where that sloughing is happening, like you said, by 4 years, you could have a whole new skin coat. Yep. Okay.
Skin coat is
um,
a weird.
What's that?
Skin coat is a very weird word.
It's kind of weird to hear it like that, but it's true. All right. So how does this apoptosis work? Well, you have certain genes that activate and start producing self-destructive enzymes. Those enzymes start eating up particular cells. Here's another example. Did you guys all know that in the womb you all had webbed fingers and toes? Yeah, you had skin between your fingers and your toes. Hello? Yes. Yes, can she finish up a few notes and then I'll send her? I just got like 2 slides left. Okay, thanks. Bye. Okay, Maddie, they need to see you about scheduling your thing for next year, but let's finish this up. Real quick, my husband's family on his mother's side, their last name is Webb. I always find this is kind of ironic. Yeah, last name is Webb. Well, some of Brian's cousins in their family, on their very last pinky toe, they actually, for whatever reason, apoptosis didn't follow through with them. And it is a genetic flaw on that side of the family. And on their last pinky toe, there was still a webbing. And so they had to physically have it cut, because sometimes there is that genetic flaw. I have a good friend that her son was born with webs between all of his fingers. And so he actually had to have surgery and just cut the skin. It's just that apoptosis didn't happen for whatever reason. They had a genetic flaw. And it's just something that's in their particular gene pool. But yeah, I just find it kind of ironic that their last name is Webb on that side of the family, and they actually have that webbed pinky toe, and they've all had it cut. I said, you should have just left it, and they could have been really good swimmers, right? Okay, anyway, so that's apoptosis. Probably the best place to put that sometimes there is that programmed cell death. Okay, uncontrolled cell growth. This is our last hurrah here. All right, anybody know what we call uncontrolled cell growth? It's an ugly word, and no one likes to hear this word. Cancer, yeah. So every year I tell people this, and then they flip out and they go home and tell their parents, I have cancer. All right, raise your hand if you have a mole. Okay, so if you have a mole, or even if you have a wart, if you have one of those, you actually have cancer. You do. Okay, but no worries, okay, because cancer has 2 forms. 2 forms, all right? And so I guess I should put up there the definition of cancer. Cancer is a disorder in which some of the body's own cells lose their ability to control growth, right? So a mole would be an example of a cancer, because it is a growth that's not normal with the normal skin. A wart, same thing, would be, an abnormally production of cells that again causes to have some sort of growth or bump, or this is what we call it, a mass, okay, or a tumor. Okay, so yes, every time I hear that word, I always think of Kindergarten Cop, that old movie with Arnold Schwarzenegger. It's so cute. If you've never seen it, it's a really old movie, but it's so cute because he's with the little kindergartners. He's like, I have a tumor. Arnold Schwarzenegger. I don't know. Anyway, so a tumor is a mass of cells that is the result of a cancer. Okay? But like I said, no worries. If you have that mole or you have that wart, just because you have cancer doesn't mean it's the bad kind. Okay? Does anybody know what we call the good kind of cancer? The word that you want to hear when you have cancer? Yes, Mia.
Benign.
Yeah, benign. If it's benign, that means that it is not harmful. Okay? That typically it means encapsulated. So benign doesn't spread because it's encapsulated. So if you want to add that word encapsulated there, that's a good word to put there, because it typically kind of stays in one central location, even though it's an abnormal growth of cells. Okay, does anybody know what the bad kind of cancer is that you don't don't wanna hear this word because it means to spread. It starts with an M.
Whatever that word is.
She's looking. Yeah, malignant. The word malignant means that it spreads. The other word, and you may wanna write this word down as well, it is another word, that starts with M. The word is metastasize. Metastasize. So if you hear the word malignant, that's never a good word to hear. If you hear the word metastasize, that's usually not a good word to hear. That means that it is continuing to spread. It's not encapsulated, whereas a benign cancer is encapsulated and it just stays in a central area. You can have some benign cancers turn into malignant cancers if you're not careful. Again, a lot of times that's based on UV radiation from the sun or tanning beds, certain lights. Okay, so, oh yeah, there's the word metastasize, I did add it. Alright, Maddie, what we're gonna do now is we're gonna take a look, I'm gonna have them watch this quick little video here, and then we are gonna take a look at some cancer cells. I don't know that we have a ton of time, but we probably are gonna grade these first, so then when we get back next time, we'll look at the cancer cells. After we take our quiz over mitosis and cell division, okay, on Thursday in here. So if you wanna go ahead and head down there, I don't wanna keep you from that. Okay, so I'm just gonna watch this quick little video. I am gonna have to turn off the lights so you can see it. I just thought it's a very old video, but I still just think it's so cool because we actually can see, cell division happening here. So give me just this, we'll let Maddie get out, otherwise she may.
Do you need our Chromebooks?
You don't need your Chromebooks anymore. No, you're good. You can shut those down, unless that's what you're taking notes on.
Share all this.
Oh, that's a good news. Is that what you wanted or no? No, not
I'm
good
getting
news.
on the embarrassing sheet.
You're like, oh, here you go. Alright, let's see if it'll pop up on here without me having to change screens. I have a bad feeling it probably isn't. Clicking yes will enable. Yes. Oh, it did. It went to a different screen, so give me a second. Uh-oh, and then the other thing is, it's not going to have the video. Scoopers, scoopers. I'm gonna have to. I have to change this for just a second so it doesn't. So it's gonna echo. How am I gonna do this, guys, at home? Cause if I turn on the volume here, it's gonna echo.
Can't they just watch it on their Chromebook?
Yeah, you guys can watch this on your Chromebooks at home, but I'm gonna have to shut off your volume at home for a second. You guys can watch this and listen at home. I apologize, because otherwise it's gonna echo and it's gonna be terrible in here. So I have to shut this off for just a second. Maybe. Okay, let's watch it. I just think this is so cool because obviously we don't have the type of microscopes where we can watch live cells. I actually tried to get some cells to see if we could see the replication, the HeLa cells, but you have to have specific types of enzyme activators, and it was way too expensive. And I bought the HeLa cells, and they were like $300, and it didn't end up working. So I had to send them back. But anyway, at least we can see it for ourselves here. Here we go. So we're gonna see cell division actually happening under a microscope. Really cool. Oh, great. You probably wanna hear the guy talk. Duh. That's forgot to do was turn that microphone on. Okay, now let's try it.
Watching a living cell divide itself into two never ceases to captivate medical researcher Gary Gorski, so he was really blown away to see it go in reverse.
I think most scientists spend long hours working in the laboratory, and every once in a while they get to see something amazing that no one else in history has ever seen before.
As we all learn in science class, cell division only goes in one direction, forward, and it cancer goes forward with a vengeance.
One of the hallmarks of cancer is uncontrolled cell division. Cancer cells have lost the ability to control their normal cell division cycle.
Gorbsky and his team at the Oklahoma Medical Research Foundation found that an experimental anti-cancer drug can rewind that cycle. As they wrote in the journal Nature, adding the drug to a cell tricked it into dividing. When the two new cells were just about to separate, the researchers removed the drug.
And the cell will reverse this process and go back to the point at which it started.
Getting that amount of precise control over cancer is likely a long way off, but Gorbsky says that cell division is such a fundamental and unique process of life.
We don't have anything else. None of our machines can do that. They can't reproduce themselves. It's just an amazing process.
That understanding it will have implications no one can even predict. I'm Brad Closen.
Okay, I wanna look at to see what date this was. 2006. Okay, so this was presented in 2006 that they had a drug that, hang on a second, I need to turn them back on. I wish this was better, but gotta. Okay, sorry about that, guys. So, anyway, what they were saying there was that there was a drug in 2006 that if it was injected into cells that were undergoing cancer, it would trick the cells not to replicate, but go back together. And so, to me, I'm like, this was 2006. It's 2021, right? That was 15 years ago. You cannot tell me that they probably have not figured out a way to help slow down cancer when that drug was doing that in 2006. Now, cancer is, not just one cell, there's several cells. So to actually go in and inject this chemical into all of those cells, that would be tedious. I don't know that they could even do that. But even if they could put it into the bloodstream where it could be delivered, you would think that that would help slow down. And I'm sure that there are those experimental drugs like these that they have out there that they're trying to utilize to see if those things would actually work. But I just think it's really cool that was there 15 years ago. And, I'm sure it's come a long way. But just kind of cool, what I really liked about that was you guys could see the anaphase there at the end was so, deliberate. You could really see it. It looked like the little fingers, as they're being pulled apart. I really just thought that was kind of cool. But then to also see it go in reverse was really amazing as well. Okay, so anyway, what we're going to be doing, and I guess we're not probably going to have enough time because we're at 50 right now, so we have 8 minutes. But I'm going to go ahead and just pull this up for you so you know what we're going to do. So what we're going to take a look at is some cancer cells. I have some cancer cells versus normal cells of various body parts. Of the lung, the liver, the ovary, I've got different things. So what I am going to have you guys look at is I'm going to have you look at the normal cells and then I'm going to have you look at the cancerous cells. Okay? So what I'll probably do, if I can, I will try my very best. So you can continue this tomorrow at home. I'll go ahead and have the slides up at home of the normal cells. So that when you come in next time, you can look at the cancerous cells under the microscope for yourselves. So you can go ahead and draw those normal cells out on your own at home. For the remainder of the period, we're going to quickly grade these labs though. I think that'll probably be our best bet. And then tomorrow, those guys will grade theirs when they come in, and then I'll have them, draw the normal versus the cancerous. So we'll just be a little bit behind in here. Like I said with the labs, that's the hardest part about trying to stay day to day is it's hard to do that. So anyway, these are some of the things that you're gonna answer and do for our small little lab with these cancer cells. Okay, so anyway, those of you at home, you guys are free to log off. You guys here, let's grade these real quick.